Welcome back to the webinar series that is sponsored by the Cure Parkinson's Trust, Parkinson's Movement, and Journal of Parkinson's Disease. So as you know, this is a quarterly webinar series. My name is Patrick Brunden. I am at the Van Andel Institute in Grand Rapids, Michigan. Actually, right now, I am in our attic. Who could have imagined at our most recent webinar about 3 months ago that we would all be sitting at home and working from our attics? Well, that is the reality, but Parkinson's research and Parkinson's care doesn't stop because we have a pandemic. So we are going to continue discussing some interesting issues in Parkinson's research. And today, the theme is neurotrophic factors in Parkinson's disease. And we have an outstanding panel that will be doing the discussing, and I will be moderating. I also welcome questions from all of you. You can see that there is a little field in the Zoom where you can write questions, and I'll be filtering through these, trying to answer as many as possible. Those that we can't answer right now, we may answer via email or on a web page later. The whole webinar will be on YouTube, hopefully tomorrow, so you can listen and watch again if there were parts that you missed. So let me begin by introducing our panelists. I'm going to start with the wife of the late and amazing person, Tom Isaacs. I think Tom doesn't really need an introduction, and nor does Lyndsey either, actually. So I think many of you have come across Lyndsey in the world of Cure Parkinson's Trust. And Tom Isaacs was an amazing Parkinson advocate, but he also had a vested interest in neurotrophic factors in Parkinson's. And Lyndsey might want to say a word about this. Hi, Lyndsey.
Hello, everybody, and thank you for allowing me to attend this panel. As Patrick said, I was Tom's wife for 16.5 years, and Tom was the driving force behind getting the GDNF trial going. It was really one of the reasons why he set up the Cure Parkinson's Trust to begin with. And since his death, I have become a trustee of the charity, and we continue to look into other neurotrophic factors, etc. Thank you.
Thank you, Lyndsey. And our second panelist is Dr. Mart Saarma, who is in Helsinki, Finland. And maybe you could tell us a little bit about your background in relation to neurotrophic factors.
Thank you very much. Hello, everybody. I am Mart Saarma from Helsinki. So I have been working with GDNF for quite many years, mostly interested in the signaling and the receptors, how these factors act on nerve cells, and more recently working on the biology of these factors and the receptors. We have also recently discovered a completely new trophic factor we call CDNF, and have taken these factors to the clinical trials. To do that, I, together with colleagues, organized a company called Untitled Pharma, and I'm one of the shareholders of that company.
Thank you, Mart. Excellent. And then I'll introduce you to our third panelist. That is Dr. Howard Federoff, who is in California in the United States. And Howard will tell us, I think, that he's currently the CEO of Aspen Neuroscience, but has done many other things in Parkinson's research also prior to that. Howard, welcome.
Good morning, everyone, and thank you for having me. It's a pleasure to be here. So my background is as a translational and clinical person. I have been involved in the development of several different approaches for Parkinson's, and those led to the development and founding of companies such as MedGenesis and also Brain Neurotherapy Bio. Prosecuting, respectively, GDNF protein delivery. I'm sorry, I have some ambient noise here. I have planes and helicopters flying around. Prosecuting GDNF protein delivery, and we'll talk more about that at length, but also the gene delivery of GDNF using an adeno-associated viral vector. With respect to Aspen, I'm privileged to be leading an effort to develop autologous stem cell therapy transplant for Parkinson's disease. And I think that the discussion today will be great because it focuses on really the key types of disease-modifying therapies that are in clinical development. And I've seen the questions, and thanks so many of you for asking very poignant and important questions.
Thank you, Howard. So just to give everybody a sense of how we are going to structure the webinar, at least how I think we're going to structure it. You never know which questions will be the most discussed ones. But the idea is that we'll start by discussing a little bit on the types of growth factors that exist and their names and how they differ biology-wise, just briefly. And then we're going to discuss how one can deliver the growth factors to the brain, the different approaches using pumps or injections or gene therapy. And then we will go through the different clinical trials that have been conducted over the years and try to understand how they were different and what the outcomes were. So I thought maybe Mart could start by explaining 3 growth factors we'll talk a lot about: GDNF, nurturin, and CDNF. How are they related? How are they different? And how have they been studied in animal models?
So I would like to start by reminding you about a very great name in the history of neurotrophic factors, a brave lady, Rita Levi-Montalcini. She actually was a lady who discovered growth factors, and it turned out that nerve growth factor, a protein which acts on neurons, was the first one. So today we will discuss 3 factors: GDNF and nurturin. These are the proteins which are the members of the same family. They actually work quite similarly. And then with the CDNF, which is a completely different factor. So how do they work? So in the brain, these factors are synthesized and secreted mostly by the neurons. So they come out from the neurons, and then they bind to the receptors, to the antenna on different neurons, and in the context of Parkinson's disease, to the receptors on the surface of dopamine neurons. And by binding to these antennas, they send chemical signals into the neurons, which tell the neurons, stay alive. We have quite nice genetic evidence that if we make animals which lack these trophic factors, the neurons start to die. So we have in vivo evidence that these factors are really important for the maintenance and for the life of these neurons. I should say that GDNF and nurturin do not act only on dopamine neurons. They act also on other neurons, but they have very prominent effects on dopamine neurons. CDNF is very, very different. Surprisingly, it is an intracellular protein which is secreted only when neurons have trouble. And they, in normal neurons, they are guarding that the neurons are doing well. They are regulating the so-called protein homeostasis. And if protein starts to aggregate, like in Alzheimer's disease or Parkinson's disease, then these proteins are activated. And they are cleaning the neurons from aggregated proteins. So they act similarly with GDNF and nurturin in a sense that they keep neurons alive, but they work also differently because they clean neurons from aggregates. So based on these wonderful properties to protect dopamine neurons and keep them alive, they have been tested in various animal models of Parkinson's disease. The work started in mid-60s with GDNF in several labs in US, Europe, showed that GDNF.
I think you accidentally said 60s, but the 90s.
Sorry, 90s, yes, sorry, yes, thank you. The GDNF was shown to be protective in several neurotoxin models of Parkinson's disease. These are the models where you inject animals some neurotoxin, which very specifically destroys dopamine neurons, and then you test whether your factor can protect and repair these injured neurons. So with neurotoxin models, GDNF nurturing and also CDNF work very well, but not absolutely well, because if you do a very severe injury, when you kill more than 80% of the dopamine neurons, then these factors don't work anymore at all.
Could I ask, when you say they do well, is the primary effect that they prevent cells from dying when they're in the process of dying, or is the primary effect that they have neighboring cells that are still alive sprouting new synaptic contacts?
Right. I think these growth factors have three major effects. The first effect is that they protect living cells from degeneration. So they send the survival signal to the neurons. The second, they actually regenerate the axons, the processes, and stimulate the formation of the synapses. And the third, they also activate the dopamine synthesis and dopamine release and uptake. So they brush up the neuron functions. So now when we come to another model, which more recently has been tested, the so-called alpha-synuclein model, then I think the data what we have with the GDNF show that GDNF is not rescuing dopamine neurons in those animals where alpha-synuclein has been overexpressed by the viral
Maybe we
vectors.
could, sorry again for interrupting you, Mart, just explain
Yes, sure.
to the listeners. Alpha-synuclein is this protein that misfolds and forms clumps in Parkinson's disease. And also in very rare cases, it's a protein that's mutated and the disease is then inherited in a familial way. So clearly a protein that has some kind of important role in the disease process. And it's possible to make the animal models that Mart mentioned by using laboratory techniques to put a lot of synuclein in an animal, and then the dopamine cells don't do well, and they form these clumps that resemble Lewy bodies that are found in patients.
Yes. So we have now a couple of papers published showing that GDNF seems not to rescue dopamine neurons if they have been expressing the alpha-synuclein, but nurturing has not, to my best knowledge, tested in this model. Now comes a little bit better news. So we have very recent data with a CDNF. And I mentioned before that CDNF is mostly sitting in the cells and trying to follow that all the proteins in the cells are in a good shape. And if there are protein aggregates like alpha-synuclein, forms aggregates and forms filaments, which seem to be the bad guys. CDNF at least gives a partial protection in dopamine neurons. But of course, we have to test that also in monkeys.
So the summary there is that three growth factors have some effects that are similar when it comes to protecting against toxin models. But when it comes to the protein aggregation, CDNF shows some benefit, whereas nurturin and GDNF do not really show that. And then the big question, of course, is how relevant are these animal models? And I'm one person who works with them, and we know that they all have some strengths and they all have some weaknesses. And it's difficult to truly predict which model is going to translate into an effect or something that you can see in a patient that they're analogous. So that's a great overview. Could I just also add that Rita Levi-Montalcini went on to win the Nobel Prize with Victor Hamburger, which Mart, of course, knows, but I think all our listeners should hear that. Quite an amazing lady who lived beyond the age of 100 and was sharp as crystal in her mind for a very, very long time and very active in scientific debate. And she did some of her research during the Second World War in a basement hidden away. Just a remarkable person. So is there anything you want to add to this, Mart, or do the other panelists want to add something to these three growth factors?
So maybe I could add, just for the better understanding of the later discussion on the clinical trials, and that is that these growth factors, when they are made by the nerve cells in the brain, are not secreted all the time, but their secretion and availability for the neurons is regulated by neuronal activity. Which is to say that just pumping a growth factor constantly into the brain is not what happens in the brain. Brain secretes these growth factors in a strictly regulated manner. And this is one thing what we should really take into account when we design clinical trials.
That's a great statement, Martin. It's a great segue for me to bring in Howard and maybe ask Howard the question, how can these growth factors be delivered to the brain? What are the different ways that they can be injected or expressed, and where should they be in the brain in Parkinson's disease?
Right, so let me just reinforce a point and extend what Mart had said. I think in the normal brain, the non-Parkinsonian brain, we know that activity is very important in terms of the regulation of release in order to effectively promote trophic function. We know far less about what happens in the Parkinsonian brain and whether that applies equally well, but it's a very important point with regard to extending them into the clinic. So how can one deliver these? There have been a variety of different approaches. Probably the first that has been studied most extensively to date has been the direct delivery of the protein, usually involving some type of cannula into a part of the brain where there is extensive dopaminergic terminal degeneration, the putamen. The delivery has been done with protein, and this is another point that I'm sure Mart will make as well, that is not biosynthesized in the same way that it would be in the brain, but rather it is being produced in another environment where some of the features that relate to the protein, its folding, and its bioactivity are very important. And so when one delivers protein to the brain, a key question, as Patrick said, is where do you put it? So that part of the putamen that lies posterior to the connection between the left and the right hemispheres. The anterior commissure is important to think about. There is a gradient, in fact, described by many, including some of those who are currently attending this meeting, such as Dr. Cordova and others. And it would suggest that the delivery to the volume where there is the most amount of denervation is the best way to think about the potential of these growth factors to act on those terminals that still are present, even though they still may be sick and ineffective in signaling both retrogradely and anterogradely. And so the protein...
Sorry to interrupt you, Howard. Could I ask a question there? So 7 years ago, you mentioned Jeff Cordova. He published a paper demonstrating that in people who'd had Parkinson's disease for about 5 years, the terminals, the dopamine neuron arborizations in the putamen were not visible anymore when one stained with a marker that showed that they could make dopamine. So the conclusion of that report was that perhaps the cells are so sick that those terminals simply are not there. They've already kind of withered away, or they are so dysfunctional that they can't produce this. What does that mean for an approach where you put a growth factor into the putamen and there's nothing there that can respond? Or should we say that perhaps those sick cells can respond?
Yeah, I think if you think about this as a state of hibernation versus the complete loss of the terminals and the loss of the cell bodies back in the midbrain is a critical distinction. So to your point, and I know Jeff's paper well as others, I think it's pretty clear that the opportunity to provide trophic support is going to probably have its greatest benefit earlier than later. And so one of the issues that has come up repeatedly in the clinical development discussions, both those that have occurred with the European regulators as well as with the US Food and Drug Administration is the willingness to allow patients to be enrolled in studies that have moderate to advanced, but yet not earlier or early to moderate Parkinson's disease. So if one looks at, and we'll talk about this in greater detail, the nature of what was conducted in the Bristol study, the one that was published about a year and a half ago, it's very clear that the category of those patients is important to understand in terms of the context of the result. But back to the delivery. So Mart mentioned that one can deliver in a sustained fashion a trophic factor. And one question is whether you need sustained delivery or whether in fact sustained delivery might cause a pharmacological phenomenon that is often seen with a variety of other both small molecules and growth factors, where the response actually could diminish in the face of sustained delivery.
So less could be more, essentially.
Less or intermittent. So that was the genesis of the design study that was pursued in the Bristol study, where the indwelling cannulae and then an exteriorized pump was used to provide GDNF intermittently, rather than in a sustained fashion, which is the way that the Amgen trial was done. So that's an important distinction, the biology of which isn't fully understood, but I would submit that intermittent delivery may be a way to avoid what could be the sustained downregulation of the response to GDNF if it were always available.
So could I also just say to the listeners, we mentioned the Bristol trial, we mentioned the Amgen trial. This might sound like code to you, but it should be very apparent in just 10, 15 minutes from now what we're referring to. Sometimes it's hard for us to talk about things without mentioning what we're going to say soon, but that's why Howard is referring to the Bristol trial being different to the Amgen trial.
Thank you for the clarification. I'll just, Mart, did you want to make a point before I go on to gene therapy?
I would like to, Howard, I would like to add to what you said, that indeed the site of delivery is crucial, because the data what we have and what has been published quite clearly shows that if we want to regenerate the axon, we need to give the growth factor to the close to the axonal tip, and then the growth factor together with receptor is taken into the cells and then transported to the cell soma, and all the way when the growth factor is traveling from the axonal tip to the cell soma, it signals. And without this signaling, axons don't regenerate. And I think this is very, very, very important. If you have patients who don't have axons anymore in the putamen, you can't regenerate them because there is no axonal transport anymore. So the second point that I would like to make is that I think also the growth factor concentration is very, very important. Unfortunately, we don't know that very well in the case of GDNF or nurturin or CDNF, but we know that for several other growth factors. And I think the broad rule seems to be if you put too much of the growth factor, the cell senses it and takes it into the cells and sends to the degradation because it's too much. So you will then occupy the cellular machinery to get rid of the excess of the growth factor. So the dose is actually very, very important.
So I've heard 4 things here. The location is important. We're talking maybe at the terminal site, the stage of the disease, perhaps earlier rather than late. The total dose seems to be important, and whether it's delivered continuously or intermittently might also be very important. So I think there's a picture emerging that this is very complex. And when one does clinical trials, it might be difficult to control all of these factors or choose the right mix of these. But go ahead, Howard, could you tell us about gene therapy? This sounds like science fiction. So we mostly talked about ways of pumping it in, but what's the trick with gene therapy?
So a lot of great work was done by many virologists that led to the creation of a set of what are called viral vectors derived from parent viruses, some of which cause human disease. Probably the most relevant for today's discussion are those that come from a type of virus called adeno-associated virus, which is described as a parvovirus, and it does rarely cause features of human clinical disease, but they are self-limited, and people, particularly children, recover from them. The development of the AAV vectors really was a major leap forward in the ability to deliver genes to a variety of different types of cells. And this work led to a number of different trials, some which involved the expression of nurturin. I mentioned Jeff Cordova, a company that was the sponsor of a number of clinical trials, including several phase 2 clinical trials have been conducted under their sponsorship. And then separately, work has been done by others, myself being a part of that effort, which was to use a adeno-associated virus that is a normal serotype found called type 2 to express GDNF within the brain of Parkinson's patients. Where it's different, just to make the point starkly, is that now the GDNF gene has been transferred into cells that can then synthesize the GDNF protein. And they therefore can modify that protein as it would normally be produced, release it, as Mart has already told you, in a manner that is likely to be relevant physiologically, and also within concentrations that are likely to be important in terms of producing the effect of the GDNF on its receptor, the protein that is known as GFR alpha 1. The gene therapy adds another complexity, which is you need to sufficiently deliver the viral vectors, which are, if you will, particles, into the space that we talked about, the putamen, so that you can have substantial amounts of coverage of that structure in order to be able to have enough GDNF that might be released and made bioavailable. One additional observation, which was really pioneered by one of my colleagues, Christoph Bankowitz, was the observation that when one takes an AAV2 GDNF vector, or for that matter, AAV2 expressing other genes just to mark how the viral vector behaves, is that the vector is able to be transported. And it's the transport of the vector from the site of installation, the putamen, to the midbrain, not immediately in the vicinity of those that die in Parkinson's, but very, very nearby, that then leads to the expression, release of GDNF that can bathe those cell bodies that are the ones that unfortunately are lost in Parkinson's, and at the same time can produce GDNF that can be released both locally, and some of that is also being released at the site that the viral vector itself was delivered within the putamen. So the distinction is really important. Just to summarize, viral vectors introduce genes and they get expressed by cells. Cellular delivery is that the protein, in this case GDNF or others, are produced outside the body and are being infused into the brain with the anticipation that they will be able to act on, in a physiologically relevant manner, the receptors that mediate their action. The second issue is one that's very important from a safety issue, which came up early in the discussions with the Food and Drug Administration, and I'm sure with the European Medicines Agency. Once an AAV vector is placed into the brain, as far as we can tell, it will be there forever. So there was a safety consideration. We've now seen more than 100 individuals in different types of clinical trials receive AAV vectors, and they are very safe. They have not.
Could I just, because we live in this pandemic era, Howard, let's just reiterate, these viruses don't typically cause disease, but also that these are genetically modified viruses, so they're not going to replicate and start spreading throughout the brain or the body.
Correct.
So that's important.
They've been totally gutted. The only thing that they can do is gain access to a cell's interior, have that gene that was brought in as the payload, then be available to be expressed by the cell, and nothing more. They cannot replicate, they cannot spread, because there is no replicative machinery in the brain that would allow them to do so.
So that's been a great overview of the whole issue of delivery. Where do you put the growth factor, what stage of the disease, dose, etc. And then the two main approaches. One is to actually use a device to inject the growth factor, and the other one is to use genetic engineering to make cells in the brain produce it and release it. So I thought we now, as we're approaching about that, we are at exactly a halfway mark, that we're going to segue now over to the clinical trials. And we've got our first three questions, and I think we'll be able to answer them later in the clinical trial discussion. They're more related to clinical issues that I can see. But I thought we'd go through the history. How did this develop? And clinically, what were the things that were positive and what were the setbacks? And Lyndsey, maybe you could tell us how you and Tom, for the first time, ran into GDNF about some 17 years ago.
Yes, so Tom, for those of you that don't know, and I don't think there's many of you, did a coastal walk. He walked around the whole coast of Britain. And on about day 2 of his walk, there was a phone call from a friend of his that said, you can stop walking, they've found a cure. And it was the first media publication of the very first Bristol 5 trial in 2002. And so Tom, on his walk, when he reached Bristol the following year in 2003, managed to meet Professor Stephen Gill on a bridge across Bristol. And he told him about his trial. And Tom, for the first time, felt he'd met somebody who thought they could cure Parkinson's. And he was so excited. And when he got back, it was really the starting point of trying to find a way to keep that trial going.
So you can say that Stephen Gill reported some very exciting results around 2003, 2004.
Yeah.
And perhaps we could say that was the beginning of the positive era of growth factor trials in Parkinson's, a lot of optimism. And maybe Martin, Howard, want to recollect what you remember from those trials and what they showed and what they led to in the second phase of studies. Go ahead, Mart. Oh, Howard, either one of you. I don't know who, both nodded, I think.
I'll add on.
So actually, I heard about the GDNF the first time probably in 1992, 93 from Barry Hoffer, just a little bit before the science paper was published. And at that time, I was mostly interested in the neurotrophic factor receptors, not so much about the clinical aspects. And I was really surprised that the growth factors was very, very similar to the TGF-beta, but apparently was not signaling through the TGF-beta receptors. So the first trial was very quickly after the first publication of the GDNF, actually before the receptors or the antennas which recognize the growth factor were published. So I know that quite well because my lab was one of the third who discovered the receptors and published in 1996. The first trial, I think, in a way, I think it was initiated maybe too quickly. And the protein was delivered into the ventricle. And they started off by moderate concentrations of the GDNF with no therapeutic effect, and then the doses were raised to quite high doses, up to 4 milligrams per patient. and then they started to see side effects. Now, retrospectively, we know that the GDNF, but nurturin in particular, are very sticky proteins. They bind to the heparin in the brain and to the chondroitin sulfates in the brain, which prevents their diffusion. So my interpretation of the first clinical trial with GDNF was that GDNF never reached dopamine neurons. And therefore, of course, you could not see any effects. So I think this is unfortunately my conclusion from this first trial.
So I can also reflect back to 1993. I think I was in Washington, D.C. at the annual Society for Neuroscience meeting, and the whole corridor of posters was filled with people talking about GDNF. It was one of the hottest topics there was at the whole meeting. And as you said, that spurred very quickly clinical trials that perhaps were done at a stage when not sufficient was known. But the optimism that came in the field, which was related to Stephen Gill's work in Bristol, that came a few years later. I don't know if you want to describe it, Howard, or Mart, you want to continue? So Stephen operated, I think it was 6 patients.
Yeah.
Direct injections into the cutaneous.
Yeah.
5 patients.
Yeah, so with, this is an important point, and I think it lays the groundwork for what I'm sure Lyndsey will talk about as well. Dr. Gill is a neurosurgeon, and he recognized that one needed to deliver the protein to the part of the brain where it was thought to be most needed. And so in what is often referred to as an open label study, so there is no control group, the protein was delivered, GDNF, and he followed these patients along with other colleagues and found that there was clearly a reported benefit on behalf of the patients using structured observations and some other measures, and also had demonstrated that there was evidence by neuroimaging of a clear biological effect of the growth factor. Eventually, the protein cessation was a decision made by those supplying it. And so Dr. Gill continued to follow those patients along with others and found that as a small open label group, there seemed to have been some sustained benefit of the earlier antecedent delivery of the GDNF protein. That got many of us very, very interested in really trying to understand whether this was now ready for prime time in terms of a more structured, well-designed and controlled study. And I think that led to the design that ultimately became part of the Amgen trial, which was a phase 2 study. And in that phase 2 study, it was clear that 2 things were important to look back on. The first is that it wasn't, to Mart's point earlier, known how much to deliver. And it was also, from a delivery perspective, while this may seem to be a simple matter, it's much more complicated that you need to deliver with a device that gets the structure bathed in the protein that you're hoping will produce a clinical benefit. So the results of that clinical trial in terms of its clinical readout was negative. And there was another issue that had been raised, which some of us had looked at a little bit more closely, which had nothing to do with the clinical trial, but rather to some non-human primates that had received very, very high levels of GDNF with the sudden termination of that, where it produced a small set of neuropathological findings, which became of concern to Amgen, partly contributing to their decision not to further continue the clinical development of GDNF.
And just to clarify, those changes were far away from the site of injection, so they were in a part of the brain called the cerebellum, and nobody really knew what this meant. But a drug company that sees potentially serious side effects will always be afraid and back off, and
as they should.
Happened. Yeah,
as they should. There was an issue that raised potential questions about patient safety. In retrospect, it was clear that the device that was used to deliver produced too little growth factor because it didn't spread given the nature of the device and the way it was delivered. And so much of what then propelled a re-examination of this was the rationale behind the study design that MedGenesis put forward as the sponsor that was conducted by Dr. Gill and Dr. Alan Wan at Bristol.
But before we get there, I thought maybe Mart could tell us a little bit about the gene therapy trials that sort of developed in parallel with the company Serogene, the one that we heard mentioned that Jeff Cordova was involved in. They took this other growth factor nurturin and put it in an AAV vector and started doing clinical trials. Obviously having done animal experiments showing efficacy before doing the clinical trials. But these were injected either into the putamen or both the putamen and the substantia nigra. And I think I can say that I remember us all in the field being very hopeful and optimistic that this might be a great way to deliver the protein and therefore we could hope there'd be benefit. But the outcome of all these trials were essentially that they did not meet their primary endpoints. And Mart, why do you think that was the case?
So I would like to add what Howard said about the phase 2 trial with the GDNF protein. There was another problem, and that was that in some of the patients, antibodies to GDNF were detected. And that was a great surprise for me because when you study immunology, you know that antibodies are produced by the beta cells. And beta cells are located in the periphery. So if you inject into the brain, you can't get antibodies. So obviously the conclusion is that there was a constant leakage of the GDNF, because if there was a single leakage, everyone who has produced antibodies know that if you take protein, this is very similar to your body. You don't get easily antibodies. You have to put them into the adjuvants, etc., etc. So there was a problem with the leakage.
The patients' bodies were recognizing the treatment as a foreign thing and was trying to mount an immunological response to it.
So meaning that some of the protein was pumped more or less constantly to the bloodstream. So coming to the nurturing clinical trials, I think it was very good that nurturing was taken to the clinical trials, although nurturing compared to GDNF is a little bit more complicated protein because it's even more sticky than GDNF. Its diffusion in the brain is even more limited. And then it primarily binds to another core receptor, which presence in the human brain at that time was not so clear, but it can also signal through GDNF receptor. So the results were actually when they first put the virus into the putamen, they saw a difference in the patient behavior. It was even statistically significant, but clinically very modest. And then they, in 2 autopsy studies, they noticed that nurturing didn't diffuse well. So they decided to put in the follow-up study nurturing virus in addition to the putamen close to the nigra. But this also didn't significantly improve the situation. So there is a recent paper by Jeff Cordova. It's a very beautiful paper published in Brain, which analyzes now post-mortem tissue of the patients. And these are very, very valuable data. For the first time, he also shows not just the protein diffusion, which is also important, but it doesn't tell, is there enough protein to activate the receptor? So now for the first time, Jeff shows also in which area nurturing activates the receptors. And actually the area where this receptor is activated is smaller than the protein. So summa summarum, I think with the nurturing, if one asks, would one repeat these experiments in clinical trials? My recommendation would be not to do that, because the pharmacokinetic properties of the nurturing protein are not very good. So I would really look for mutants of nurturing or GDNF which have better diffusion properties, better solubility, better stability, and maybe also improved activity. Well, if we look to the neighboring field, diabetes, and look at insulin, so what we buy, the insulin what we buy, it has been heavily genetically engineered. In our field, GDNF and nurturing what we have used are exactly the ones what nature has made them. We have not improved them. But there is a long way and a lot of possibilities to make better GDNF and better nurturing. And there are some animal studies now with improved nurturing published.
So you're saying that there's more to do on the technology front, not just with regard to the delivery and the dosing, but also with actually the exact
Absolutely.
protein that's injected. But nurturing,
Absolutely.
the story ended, the company Serogene is no more, and they couldn't continue. There was then a gene therapy trial with GDNF. Maybe you could just say 2 sentences about that, Howard, because I want us to move on to the Bristol trial. We have about 15 minutes to go, but this was a parallel also development to the Serogene nurturing.
So very briefly, a group that were academics had received permission to use the resources of the intramural program at the NIH by then NINDS director Story Landis. A phase 1 clinical trial was initially proposed that the FDA cleared as a safety and tolerability study. It was to actually enroll 18 patients. There were going to be 6 patients in 4 different cohorts. At the end of the trial, it was clear after the 13th patient was operated on that there was safety and tolerability. And it was on that basis that the investigators believed that it was worthwhile considering extending that work into what is now a phase 1B to a phase 2A. So that was
Explain the difference, Howard.
Yes, so the issue with regard to a phase 1 study, it's almost always to demonstrate safety and tolerability of a drug, including a protein or a gene therapy. And that's what was done in the 13 patients that were part of the phase 1 study that was done intramurally at the NIH. That would be considered a phase 1A study. A phase 1B study would be intended to expand those results, again, assessing safety and tolerability, but begin perhaps to evaluate other features, which could include further dose finding and maybe even other methods that would improve either the manufacturing of what is called the test article or the drug investigational agent, in this case, a gene therapy, and again, demonstrate safety, tolerability, and then enable the first study, a phase 2 study, which is where the earliest signs of what could be equated with clinical benefit could be assessed. So that is work that is unfolding currently, and I'm sure in the next years to come, there will be data reported.
So GDNF, gene therapy, is still alive and kicking and being investigated. But Lyndsey, tell me then what happened when you and Tom reconnected with Steve and Jill, and there was the trial that was reported about a year ago. How did that start?
So Tom continued to be the driving force behind trying to get GDNF back into trial. I know that the Cure Parkinson's Trust funded Steve Gill to get to help with the new delivery mechanism, the convection enhanced delivery mechanism. And in 2012, Tom was one of the first 6 in the safety trial for the new Bristol GDNF trial using this new delivery mechanism that then continued until 2017 when the results were published last year in 2019. Unfortunately, as we all know, the didn't meet its endpoint. The outcome was not what was expected from a lot of the participants. And there was also a documentary that was filmed following 5 of the participants through the trial and following them through. So there was a 9 months placebo, double-blinded, and then it became open and everyone went on the drug.
So that's very interesting and important because we know in Parkinson's disease that the placebo effect is profound with any treatment. And that's not a bad thing. If you give a pill and you say you're going to feel better or do better, people will do better with almost any disease, and it's particularly evident in Parkinson's. And it's also known that if there's a surgical intervention, placebo effects are even greater because there's even greater expectation in the patient that this is something very, very major that's being done. So let's talk a little bit about the outcome of this Bristol trial. There's no doubt it didn't meet its primary endpoint, and therefore it wasn't efficacious. And yet many individuals who participated in the trial perceived that they had gotten a lot better. And if you watch the documentary, of course, there are stark differences. If you see the video footage of these people, what does that mean? I'll give this word to anyone. What is it really? And we have questions here from individuals who say that why is there no interest in a follow-up study if the Bristol participants might have improved clinically in terms of and looking at the different groups that were operated there. So has the final word been said in the Bristol trial, or is there a reason to go back and dissect it in an even more fine way, although that's not allowed if you follow the real rules of clinical trials? But is there something we can learn for the future from looking more carefully at the data? Who wants to answer that? That's a very difficult question.
I'm happy.
That's hard.
My strong belief is that we can learn a lot from it at many levels. First, we mentioned this at the very beginning, you did, Patrick, that some ways to look at neuroimaging allow us to explore the biology of what a drug like GDNF might be doing. The strongest readout of that trial was using a way to look at the capacity of the dopamine terminals to actually manufacture dopamine using an imaging agent. And I would say, so strikingly, was there a significant and large increase in patients who had received the drug in a randomized setting that had a very, very big and highly significant increase. That's a biological readout that I think cannot be ignored. The second thing, which is perhaps a conclusion that I share with many, is that I think the 9-month time to look at clinical development was inadequate. I've come to believe from looking at all that has been done today that the very first phase of response to a trophic factor such as GDNF and maybe others is going to be the reactivation of the capacity to function as a dopamine neuron, such as what the neuroimaging readout showed. But then later, one has to have the full elaboration of circuit reconstruction that would then be equated with clinical readouts. And then finally, and I'd love to hear Lyndsey's impression, I think that the importance of the patient-reported outcomes and their appropriate weighting in the conduct of a clinical trial is vitally important to understand whether the trial is a success or a failure. And my strong belief is that if those are inadequately accounted for, as I believe they were in the primary endpoint for the Bristol trial, then one still has reached a conclusion based on agreement with the regulatory agency that you have failed to meet the endpoint. Yet I believe that if one had appropriately weighted other readouts, including the patient-reported outcomes, the trial would not be viewed as such.
So Lyndsey, what do you say about that?
Yeah, I totally agree with Howard. It is one of the things that we have been discussing as a group that I'm part of people and their families that were part of that trial. because when Tom was going for his assessments, he would be practicing everything. We would do everything last minute, the diaries. And I think one of the great things that has come out of this trial, there's been two things really, and Tom always felt that the measuring of Parkinson's was exceptionally poor in relation to everyday living, you know, stuff. Have I got time to give a quick quote from one of his speeches he gave?
Absolutely.
Yeah.
Go ahead.
Yeah, so he said, it was a speech that he gave at the last meeting of all the participants of the trial in March 2017. And he said, some of the Parkinson's assessments do have their limitations. One of the questions I always liked was, over the past few weeks, have you been more or less indecisive? To which there is only one reasonable answer, and that is, I'm not sure. So
I think that is a classic Tom Isaacs answer.
yes. So, but what has come out of it with a group of people, Dr. Moon was involved, was a new scoring method called PD Core, which they're trying to hopefully get taken up for further trials. And it's the PD comprehensive response, and it's looking objectively assessing motor examination, but also with 2 patient-reported outcome measures that reflect day-to-day reality of how much time a patient spends off and the impact on motor function in daily life when in the off state. And the other thing that's come out of the trial is a project called the Sphere House, which is a smart house where we are funding some PD sensors, which will hopefully capture day-to-day living in a much more subjective manner. So, yeah.
It's a very good point. So I think what has been said is that imaging indicates there is some biological signal. Maybe the treatment duration was too short. Maybe there's heterogeneity between patients. I should strike out the word maybe. There is heterogeneity between people affected with Parkinson's, and maybe there are those that are more likely to respond, depending also on the stage of the disease that they're in. But finally, as so eloquently described here by Lyndsey, perhaps we're measuring the wrong things. But all of these are questions, right? So I don't think we can say that GDNF works, not at all. We can't say that based on this trial. But perhaps we should also be humble and say that we're not sure if one changed the protocol, if it would work instead. And we have the AAV GDNF trial ongoing, and then we have the CDNF trial that Mart mentioned at the beginning, which is ongoing now with a different protein, as you heard, another growth factor that also potentially could work. I just want to highlight a question that I got here. Somebody wanted to know if you've had PD for a long time. This individual was diagnosed 16 years ago and was a GDNF Bristol participant. Are you beyond help? Of course, the answer is never you are beyond help. Of course not. You never know what science is going to bring. Based on what's been said today, however, growth factors might not be the best way forward if the disease is advanced. That's a guess, but we don't know for sure. You heard at the beginning that the cells that are supposed to respond to the growth factor must at least have their axons or their arms out there, probably, for them to be able to respond. But we don't know that for sure either. Maybe CDNF will be better in those instances. So we only have a minute to go, Mart, or actually 30 seconds for you. Tell me, when will we know more about the CDNF trial?
Definitely, yes. I would like to say that I think we learned a lot from this last GDNF trial. And we can say that this was a phase 2 clinical trial with GDNF, which had no technical problems. And there is a very clear progress from the first trial to the third one. And we can identify also in this trial several points where we can improve. For example, we can, instead of bacterial GDNF, use mammalian GDNF. I think the doses used for this trial are very, very low. The higher doses can be used. And of course, as you mentioned, the average age of the patients who were treated in this trial they had more than 10 years of Parkinson's disease. Maybe very little fibers left in the putamen. So taking mammalian protein with higher concentration and treating a little bit earlier stage patients may make a very big difference.
So I think, to summarize the whole webinar, I think we've illustrated that growth factor biology is complex. And it's not just taking growth factor and injecting it somehow in the brain and there'll be a wonderful effect. You have to consider many technical and biological factors. While there have been encouraging results on the way right now, the most recent trial wasn't the wonderful, perfect outcome. In fact, it was a negative outcome. But it doesn't mean one should give up. And I think, as we've also heard, there are many things that can be improved. The time perspective for this, it's long. We're talking here about a 25-year history of this growth factor, and still we don't have all the pieces in place. If it ever were to work, it's going to be another 5, 10 years, as we always say, another 5, 10 years. That's what we always say when we don't know how long it's going to take. So, cautiously optimistic. Is that a fair conclusion of this? That it's a challenging area. We should be cautiously optimistic. With that, I want to wrap up, and I see there are some questions we couldn't answer live. We had a great discussion. I want to thank the panelists and those that we didn't answer those questions. We tried to get back to you on the web or some way to give you feedback on those questions. And the next webinar will be in about 3 months from now. We welcome suggestions for topics. And who knows where we will be sitting. Will we still be in the attic or will we be in our offices? I hope back in the office. Thank you everyone for listening. Bye-bye.